Okay, in this video, I'm going to explain the difference in the action potentials between autorhythmic cells of the heart and pumping cells of the heart. So recall that the autorhythmic cells make up about 1% of the myocardial cells, and that the autorhythmic cells that are in the sinoatrial node generate action potentials the fastest, and these are the ones that actually generate the rhythm of the heart. The pumping cells, also known as contractile cells, are the ones that have lots of actin and myosin in them and do the work of the heart in terms of contracting and propelling the blood through the body. So let's take a look first at the autorhythmic cell, and you'll notice that we have the y-axis labeled in millivolts and the x-axis labeled in seconds for time, and that's going to be the same as for pumping cells. And that notice that for each action potential we have 4 phases labeled, 1 through 4, and so for each blank we will discuss what's going on in each of those phases. So in step 1, you can see that we have what looks like a graded potential, and it actually is a graded potential, and in the heart it's actually called a pacemaker potential. And so this is caused by after hyperpolarization, and that happens actually at the end of the action potential, so it's kind of like a chicken and the egg thing. So we'll come back to the end afterwards, but just accept that hyperpolarization causes a sodium leak through some channels that are called funny sodium channels, and these open in response to hyperpolarization. So we end up getting increased permeability to sodium that causes this graded depolarization to happen. So during this phase here, we have some increased permeability to sodium that causes us to start having this graded potential. Well, there's also some calcium channels that are sort of slow to open and close, and these are called slow voltage gated calcium channels, and towards the end of the action potential here they open, which further helps to bring this membrane potential to threshold. So I'm going to add increased permeability to calcium. So then we reach threshold. So threshold is the dashed line right here. So I guess we could label that. And at threshold, then we have the rapid depolarization phase of the action potential. And that's caused by not sodium channels opening, which is what we're used to seeing, but a rapid increase in permeability to calcium. So this is kind of different. So in the membrane, instead of rapid voltage gated sodium channels, we have rapid voltage gated calcium channels. So for this we have increased permeability to calcium here, which causes our rapid depolarization. And then the calcium channels close, and for repolarization what we're going to see is increased permeability to, as you probably guessed it, potassium, and of course a corresponding decrease in permeability to calcium. I'll just write that there because they close. So the repolarization and after hyperpolarization, so going a little bit lower, is due to the opening of voltage-gated potassium and closing of voltage-gated calcium channels. That after hyperpolarization is what causes the opening of those funny sodium channels in the first place. I kind of skipped over step 3 because it's not so important for these autorhythmic cells, but we're just very briefly depolarized at the top. We're going to talk more about that step for our pumping cells. So for pumping cells, you'll notice that we don't have a graded depolarization to threshold. In fact, we're at baseline, oops, and all of a sudden we have our rapid depolarization phase. And that's because the action potential comes directly through gap junctions from another cell, either from an autorhythmic cell or from another pumping cell. So we don't have that graded potential there. And the rapid depolarization phase is caused not by calcium channels opening like in the autorhythmic cells, but by opening of rapid voltage gated sodium channels. So just like the typical neurons that we've seen before, or with muscle cells, we've got increased permeability to sodium. So that causes our rapid depolarization phase. And then we lead into this depolarization plateau. And so this is something different. And so what we see here is kind of a flattening out or delayed repolarization. And that happens because initially there's a brief increase in permeability to potassium. So right here we have an increase permeability to potassium, and then those potassium channels close again. And then to hold this membrane depolarized, we open some calcium channels. So we see an increased permeability to calcium, and of course the sodium channels have closed. And so this increased permeability to calcium holds the cell depolarized, and then we end up repolarizing by closing the calcium channels, so decreased permeability to calcium and increased permeability to potassium. So that's been the one constant for all of our excitable cells so far, is they repolarize because of increased permeability to potassium. So why do these pumping cells stay depolarized for so long? Well, if we look at our action potential of our pumping cell, so we have our resting membrane potential, and all of a sudden the action potential comes along through a gap junction from another cell, and we have our brief opening of potassium channels, and they close again, and then our depolarization plateau due to opening of voltage-gated calcium channels, and then those calcium channels close, potassium channels open, and we repolarize and are back to resting membrane potential. So we have increased permeability to sodium, increased permeability to calcium, and increased permeability to potassium here. Well, if we were to overlay a picture of the force of contraction of the cardiac muscle, what we would see is the muscle is relaxed, 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 relaxed. Now it receives action potentials, it contracts, and then it relaxes again. That's like a muscle twitch. So this blue line is force, and the green line is membrane potential. And what you can see is that the membrane potential doesn't finish repolarizing until the muscle's almost completely relaxed again. And what that means is that you couldn't stimulate another action potential to happen until the heart was almost completely relaxed. And this is really important because when does the heart fill with blood? It fills with blood while it's relaxing. So if you could put the heart into tetanus by keeping it contracted, then you wouldn't be able to allow the heart to relax and fill with blood and then contract and squeeze the blood out. So just like ventilating your lungs requires the alternate contraction and relaxation of skeletal muscle, pumping blood through your heart requires the heart to relax and fill with blood, and then contract and squeeze the blood out, then relax and fill with blood again. If you recall with skeletal muscle, you could totally put skeletal muscle into tetanus by repeatedly stimulating it because the action potential was over before the muscle even started to twitch. So this long refractory period in cardiac muscle is really important because it prevents tetanus in the heart. So refractory period prevents tetanus and keeps the blood flowing, or pumping, I should say. Keeps blood pumping, which is important for life.